Today's Thinking Thursday is going to be about both Henrietta Lacks and HeLa cells, and I'll explain later why I'm treating those things separately, despite the fact that those stories come from the same person. Henrietta Lacks was born Loretta Pleasant on the 1st of August 1920. Now, I don't know why she went by Henrietta rather than Loretta, but I do know why she went by Lacks rather than Pleasant. Her father was Johnny Pleasant and her mother was Eliza Pleasant, but her maiden name was Eliza Lacks. When Eliza was giving birth to her 10th child, she unfortunately passed away in childbirth. This was when Henrietta was 4 years old. And Johnny was like, wow, I have way too many kids, and started distributing his children to relatives. And Henrietta was sent to live with Tommy Lacks, her mother's father, so her grandfather. Her grandfather lived in the 2-story slave cottage that was on her white great-grandfather's land, which was a tobacco plantation. And Henrietta actually started working on the tobacco plantation, and she was living in this 2-story cabin with her grandfather and also her cousin. Her cousin David went on to be her husband. Henrietta actually had her first child when she was 14 years old, and she eventually had 5 children with David. But her story was cut short when she was only 31. In the end of 1950, she'd been experiencing really bad abdominal cramps, but she'd kind of been passing them off as like period things, when it turned out that actually this was cervical cancer. In the beginning of 1951, she went to a charitable hospital to be checked out, and she had a biopsy taken in the spring of 1951. She had one biopsy taken of the healthy cell of her cervix and one taken of the tumorous cell in her cervix, and was indeed diagnosed with cervical cancer. However, what she didn't know was that the tumor cells were sent off to a laboratory where they would continue to grow. Something very special was discovered with her cells, and that was that they are hypertriploid, and that means that they have more chromosomes than normal healthy cells. Normal cells have 46 chromosomes, whereas hypertriploid cells have, in her case, 76 to 80 chromosomes. That's a lot more, but something else special was happening with her cells. Normal cells are very difficult to grow in a lab because they die quite easily, whereas her cells turned out to be something that we call immortalized cells. Her cells, instead of breaking off the end of the telomeres, so the telomeres are like the end of the legs of the DNA, so if you think of a double helix, you have this kind of cross of DNA. Normally, when that DNA divides to do something else, then the ends get broken off, and you get a limit to how many divisions that DNA can actually perform. Usually this limit is around 50, and that's called the Hayflick limit. However, with her cells, the telomerase was actually going and rebuilding the ends of the telomeres so that her cells can just keep dividing and dividing and dividing. To imagine this, think of a braid of hair. So you have this plait, and every time the plait comes undone, the ends break off. And so eventually you have no plait at all. It can't do anything, the hair's too short, it's gone. And this is when the cell dies. That's called apoptosis. However, with her cells, there's a magical force, telomerase, that's going to the end of the hair and rebuilding the hair so that not only does it stay the same length, but it can keep dividing. It can keep making more and more plait. This is pretty magical, and it's pretty rare. Her cells actually went on to be the most used cells in research ever. Now, hers are not the only immortalized cells. There's actually an entire encyclopedia of immortalized cells that are used in research, but hers are just the most popular. Now, these cervical cancer cells taken in 1951 have been used for everything from polio vaccines to radiation effects to zero gravity effects. They've pretty much done most things in biology, which is really cool. But this is why I'm going to treat them as separate from Henrietta. I think they have 2 separate stories. And the reason for that is because of epigenetics. Epigenetics are the changes that happen to DNA from the environment. So it's not just the characteristics that were given to you originally by your parents, but the characteristics that come from the environment. For example, if you have twins who share exactly the same DNA in the womb, they won't end up having the same DNA exactly at the end of their life because they'll have environmental epigenetic factors that change their DNA over time. For example, if you have one twin who lives at zero gravity, their epigenetic effects are going to be very different to a twin that lives with gravity, because the density of the bone is going to have to adapt in the zero gravity twin versus the twin that's under gravitational pressure all the time. Same as if you have one twin who is really lazy and eats terrible food, and you have another twin who's a fitness freak and eats super healthy food all the time. Those epigenetic factors are going to be playing into the DNA factors and changing those twins epigenetically over time. This is why I'm saying that the Henrietta Lacks story and the HeLa cell story are different. They're like twins. They started off with the same DNA and then epigenetically they changed. Now this is a pretty radical change because Henrietta Lacks died in 1951, whereas the cells, the HeLa cells, have continued to live on due to being cryogenically sustained in laboratories. Those are some pretty different experiences for cells to go through. So this is why I'm treating these stories as different, because genetically they are. However, Henrietta's family have claimed that they should have the right to decide what happens to those cells, because they claim that they have the right to that information. So if you think about patenting information, if Henrietta owned the information of her DNA, then that ownership of information should pass down to her family. Just the same way as if she owns a house, then her children will inherit the house. But that's not necessarily what happens. And in fact, in US law, it's pretty much the case that if you have left cells somewhere, you don't really have legal capacity over them anymore. So for example, if your hairdresser decides to make a wig out of your hair, they can then sell it and they don't have to pay you for that wig. This is a pretty hotly debated topic. And in fact, there was someone who had his spleen removed and fought for the right to have the financial benefits of the research that had been done on his spleen, because his spleen cells were being used for research and he wasn't being paid for it. And in fact, he first lost the case, then won the case, and then lost the case when it went to Supreme Court. I'll leave some information down below so that you can see that as well, but it's still something that we aren't quite decided on. What happens to your cells after you aren't using them anymore? And should you get the financial benefit if someone else is? We don't really have a solid answer. I'm someone who thinks that everyone should be an organ donor unless they've specified not to be, but that's not the way law works. You have to specify that you want to be an organ donor in order for your organs to be used for someone else. In that way, we kind of give the right over to the person, even if they're dead, to decide what's happening to their organs. But I think, actually, if you're not using them, someone else could. In the case of Henrietta, her family has actually won compensation. It didn't happen until 2023. That's when they finally got compensation. But to be fair, her family was struggling to pay for medical bills, and Henrietta's cells have earned the medical industry billions. And I can kind of tell why. The price for a single epithelial cell of low passage, so that's a cell that hasn't been used very much for doing things, goes for $968 per cell. There's a lot of cells that were taken, but these are cells that can infinitely replicate. That's really useful for the medical industry. And the thing about them is that they're incredibly resilient and they like to replicate. They're very aggressive replicators, so they're very, very valuable. Her family wasn't seeing any of the benefit, and they didn't even know about it at the time. They only found out that Henrietta's cells were being used because they were actually called to supposedly have blood tests and to see if they had any cancer themselves. I'm pretty sure that's because the medical industry wanted to know if they also had some kind of genetic mutation that meant they had cells that were super useful and resilient for reproducing in the lab. And it was Deborah, Henrietta's daughter, who kind of clocked on to the fact that there was something going on. They finally found this out, but it wasn't until 2013 that they finally had the right to decide what would happen to the cells. And that only meant that one family member could sit on a board, like a committee, to decide who gets access to the information or access to Henrietta's cells, but not really much else. And it took another 10 years for them to get any kind of compensation at all, which was after the industry had made so much money from the replication of these cells for research uses. This medical ethics problem has been quite well covered. There's books about it, there's loads of research about it, there's loads of articles about it. But one thing that really hasn't been talked about is the medical ethical bullshit that went on during Henrietta's life. I don't really understand why a spotlight hasn't been shone on it. During 1951, when Henrietta was still alive and receiving treatment, racism was rampant. This was the era of segregation, even to the point where when they took samples of Henrietta's bladder, bowels, uterus, kidneys, vagina, and ovaries, those cells were put in a freezer called the colored freezer because they had to be segregated from samples from white people. It's that level of racist. But what happened to Henrietta is so unfortunate and should be talked about more. During treatment, her kidneys began to fail, and this meant that she needed blood transfusions. However, they actually temporarily stopped her blood transfusions because she needed so many and told her that she had to replenish the blood bank deficit. How if she needs the blood transfusions? That makes no sense. Fortunately, her cousin Emmett Lacks then gathered all the male relatives of the family and they sort of boosted up the blood banks so that she could continue having transfusions. But that's not how a hospital is supposed to work. They're not supposed to say to the patient, go and figure out your own treatment. I've tried to find cases of white patients being denied transfusions at the Johns Hopkins Hospital in 1951 due to the blood banks being low, and I can't find any. Now that could be because I'm doing not enough research or that I just don't have access to those medical files, but I'm pretty sure in this era of segregation, white patients were being prioritized over black patients. Henrietta unfortunately died at the age of 31 in October 1951. I don't know whether the pause in her transfusions sped along her death, but she didn't make it through the rest of the year. What happened to her cells was that they did. They got cryogenically frozen, and then they've been used ever since. Due to the fact that they can replicate very easily and they last a lot longer than healthy cells, they've been used in so much research. Jonathan Landry and Paul Phil actually published the genetic sequence of her DNA online, and they work at the European Molecular Biology Lab in Heidelberg. Now, the family was not a fan of this, and I can understand why, because US health insurance premiums are pretty sneaky, and so I'm almost certain that those health insurance companies were going through that DNA sequence and figuring out, okay, what are you likely to get when you're older, and how can we bump up your premium because we can show that you are likely to get sick, so we're going to make it more expensive for you to have health insurance. I would also be uncomfortable with that too, but it is very useful to have that DNA sequence online because when you use those cells, they are going to gradually mutate over time. That epigenetic property of you putting them through different tests and different situations means that they're going to change slightly every time they go through some kind of division or some kind of medical research. So we need to have that original genetic sequence so that we can compare and see how it might have changed. It's also really useful because cross-contamination is a massive problem in research. So you might have liver cancer cells, which you think are uncontaminated, and this is really important because they have different properties like fat producing and things like that, which cervical cancer cells don't have. You need to understand when those cells are contaminated because if they have different properties, then they're going to have different behaviors. Every time you use a HeLa cell, it's going to mutate slightly. So for example, if you divide it, it's going to change slightly as that telomerase is creating literally new DNA ends, so lengthening the telomeres of that DNA, but also when you put it through divisions that maybe are part of a science experiment that adds something new. For example, with coronavirus, they used HeLa cells to test COVID vaccines, but they had to transduce, so add a gene into the HeLa cells that they didn't have. This gene was the ACE2. So ACE2 is the angiotensin converter enzyme 2, which is an enzyme that breaks down angiotensin. And angiotensin, I like to imagine as like tiny little grippy hands inside your cell. Now, its job is to regulate blood pressure. And what it does is it really quickly constricts blood vessels to lock in water and sodium to raise your blood pressure so that you can regulate your blood pressure, because very low blood pressure is also very dangerous. However, people with too much angiotensin have to go on ACE blockers or inhibitors, because then it means that they have hypertension or high blood pressure. ACE2 is the angiotensin converter enzyme 2, which breaks down the angiotensin, and it basically, imagine if it's like little hands that are like grip, 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 grip. It breaks them down, like snaps all their fingers off and leaves them at the surface of the cell. And these little fingers on the surface of the cell become hooks. And this is how COVID enters the cell. The angiotensin broken down into these smaller molecules that hook in or are the access route for the COVID virus. ACE2 has been shown to be a double-edged sword because, yes, it does hook in COVID virus. If you've seen those little spike proteins sticking out of the edge of a COVID image, those spike proteins are basically hooked in by the broken down molecules of the angiotensin that's gone through ACE2. And that's how the COVID really attaches to the cell, through the little broken down particles of the angiotensin that connect with those spike proteins, and then it can like attach. However, it's also been shown that ACE2 prevents severe cases of respiratory syndrome. Somehow, those little particles are preventing SARS-CoV from becoming really, really aggressive. The role of ACE2 is to break down angiotensin so that it can't aggressively inflame the cell. And when it comes to COVID, it wants to aggressively inflame the cell. And this is what ACE2 prevents. It breaks down the COVID just like it would break down the angiotensin. You have ACE1, which induces the angiotensin of the cell, so that increases the sodium and water levels of the cell, which damages the cell over time and can lead to cell death. But you have ACE2, which breaks the angiotensin down so that it can't inflame the cell too much, so that you get a balance between ACE1, that's increasing the pressure of the cell, and ACE2, that's decreasing the effects, so that you get a balanced pressure. So you need to have reasonable balance between the two. SARS-CoV incapacitates ACE2 so that it then attaches to the ACE2 molecules, which prevents them from being able to stop the angiotensin going ape and increasing the sodium and water levels of the cell to the point where the cell goes through cell death from inflammation. This is how COVID becomes deadly, because if you have enough cells that die, eventually the body actually deoxygenates, because there's not enough healthy cells to pass oxygen around the body. This is why COVID became deadly, basically because SARS-CoV attaches to the tiny little hooks on the surface of the cell that is the ACE2 broken down parts of angiotensin, stops them from being able to continue to break down further angiotensin. The ACE1 and ACE2 go out of balance, and then you have the inflammation that's caused by the angiotensin increasing and increasing and increasing until the cell dies. Over time, this accumulated effect in the body leads to not only death and deoxygenation of lots and lots of cells, but eventually the whole body. People who are on ACE inhibitors, like blood pressure medication, are actually potentially better off when it comes to COVID, because they aren't producing as much ACE1, which means that the angiotensin isn't being promoted in their cells. That's what ACE1 does. It wants to induce the production of angiotensin in the cell, and that means that when it comes to the SARS protein infection, they might actually not have so much angiotensin to worry about in their cells. So that issue of the SARS-CoV virus entangling itself with the ACE2 and incapacitating it, basically keeping it occupied so that it can't be really dealing with the angiotensin as well, isn't such a problem because there isn't so much angiotensin in the first place. There have been some studies that have shown that actually, particularly in the case of people with cardiac issues that were on these ACE inhibitors, were slightly better off. They had less severe cases of COVID and they need less hospitalization. Also, their death count was lower. I'm not saying that we should all suddenly go on statins, but I think there's more to be explored when it comes to ACE inhibitors and COVID viruses. There's going to be more viral infections like COVID. We're going to see more and more pandemics, especially as populations continue to grow but locate themselves in the same cities. So I'd be really interested to discover whether things like ACE blockers can actually prevent viruses from taking such an intense hold. I have 3 questions that I really want the internet to turn its attention to, and I don't really think that any government out there would be willing to pay for this research. So I'm gonna just put it out there and hope that some of you are interested and we can together use our interneting powers to try and answer these questions. So I want to know if there's a comparative study between HeLa cells that have been transduced with ACE2 to test COVID vaccine and cells that naturally produce ACE2 to see if the entrance behavior of SARS-CoV-19 or just its behavior within the cell is different between a cell that has been induced by a synthetic form of ACE2 to express ACE2 and a cell that naturally produces ACE2. I would like to see if there's a comparative study between the two. I would very, very much like to know if there's a difference in how the SARS virus interacts in those 2 different types of cell. Secondly, with cells that weren't HeLa cells, how can we know that they weren't cross-contaminated? The issue of cross-contamination is a hot topic in biological research because HeLa cells are so aggressive at mutating and spreading that there's so many cases of cross-contamination. Under the circumstances that research centers were under during COVID, they were given so much pressure to produce vaccines at such a fast and unethical rate that there's high likelihood that they weren't always able to consistently check back against the database for every single cell that they were using. So how do we know that there wasn't cross-contamination or some kind of mutation happening if they weren't able to take the time to consistently check back and see, oh, were there any mutations, or have we got any gene shifts, or is this just the wrong cell entirely? This was a time where biological research was having to happen at a speed where protocols were kind of having to be shortcut occasionally so that they could produce the results that they were being pushed for and paid for at the rate that they were asked to do so. And my final question is, how do you decide how much ACE2 to transduce into a HeLa cell to make it a relevant experiment for a vaccine? Every person has a different level of ACE2 in their cells, so how do we decide what the medium of mankind is? And what's the effect of a vaccine that's been tested on synthetically transduced HeLa cells that have ACE2 added to them for someone who has an ACE2 level that is nothing like the level of ACE2 that's been transduced into that cell? Is that still relevant? How do we understand the variation in mankind when we're making a vaccine? And what level of testing is done for the vast spectrum of ACE2 levels that is in mankind? How relevant is the publicized information about whether a vaccine is safe versus how diverse the human genome is? Every human has a different level of ACE2, ACE1, the angiotensin in their cells. So when we're saying that this is the effect of SARS-CoV on a cell, what we really mean is this is the effect of SARS-CoV on a cancer cell that has been induced to express a gene in a completely unnatural circumstance. We don't actually know what the behavior of SARS-CoV is like in every single person because we couldn't possibly clinically test that. The variation in mankind is too wide. So all we're ever looking for is a medium. But my question is this the right medium? Is this the mean of mankind? Is this the average of mankind that we're looking at here? Because we have a cell that is not like a healthy cell. It replicates completely abnormally. It doesn't contain the specific gene that we were looking to assess by using it in the first place. And more than that, is it right to consistently use HeLa cells, even though they mutate over time? Should we not be donating into cell banks so that we can have fresh cells from actual humans that are healthy to do research on? This would require humans to actually donate healthy cells for this kind of testing, but I think everyone can spare a few cells. Now, sometimes this means stem cells, and period blood has been used to do research, and it's really, really useful. So there might be a case for period blood being donated for the sake of research. When we are looking at research for the whole of mankind, we have to have a wide variety of cells. And most research is being done on cancer cells, not healthy cells. So I would like to push for period blood being donated to research centers so that we can get a wider variety of cells being donated to these centers to create a more accurate picture of mankind. Instead of using this small bank of these infinitely replicating cells, if we have a more diverse population of cells to use, we can get a more accurate picture of how these vaccines or these effects like zero gravity work on a wide variety of cells. This represents humanity more accurately rather than cells that just come from one lady a very long time ago. I would like to propose that period blood is donated to research centers so that there's a wider variety of healthy human cells to represent the human genome when it comes to research. I think that it's time that we put aside the idea that just having cells that replicate really, really fast and really, really aggressively is the only way to research mankind. I don't know how to make this take off, but I think it's a really good idea to explore. Let me know if you think so too.